Assalamualaikum. So this thing, starting from this thing, it will be my turn to take over this class. So basically, I will be covering on the proteomics part. So for this thing, okay, we will be continuing with the proteomics. All right. So just a basic or kind of, I'm not going to delve too much on this topic, even though probably from your notes you will see that it's actually a bit lengthy, but those are the basic stuff you have learned it, and I think that the year would have covered it in a class as well. So I'm going to go briefly into it if you are interested, okay, when we have an online discussion later on, so you can ask me anything, or you can just post your question underneath this video. So proteomics, okay, so whenever you see omics, okay, towards the end of any word kind of like genome, transcriptome, proteome, metabolome, okay, but with a mix or omics at the end, it means that it is a study of. So proteomics means it is a study of protein. So regardless of whether it's a study of sequence, structure, function, design, or modeling, anything about protein, either one of them or every one of them, okay, or a combination. So a lot of time proteomics is complementary to transcriptomics, because transcriptomics is most of the time the least studied among the omics as compared to genomics, proteomics, and metabolomics. So proteomics involve molecular biology and of course bioinformatics. And as you're well aware that this course is basically a lot about bioinformatics. Now, protein is basically, we know that it is something which is fundamental, and basically every organism would produce protein, and it's actually an essential part and a part of our everyday life. So it provides structural support, storage, transport of other substances in the cellular signaling, movement, defense against foreign substances, and many more. So apart from that, enzymes, there are lots of main enzymes, okay, which are protein in a cell, and they function to regulate metabolism, and this is done by selectively accelerating chemical reaction to help in our bodily function. So protein comprised of amino acids. So we know that there are 20 basic amino acids with fundamental structure, and the only difference with this protein is basically at the side chain, which is the R group. And amino acids are usually basically denoted by 3-letter and 1-letter abbreviation. For example, methionine, MET, or M. So basically, it also, or the 3-letter is also because it is comprised of 3 nucleotides, okay, if you were to translate it from DNA or RNA. So the polymer of proteins are called polypeptides. So when you have a series of amino acids joined together, they form polypeptides. And a protein consists of one or more polypeptides folded and coiled into specific conformation, which we will see on the structure in a bit. So amino acids, basically, we know that it has 4 components. So your 4 components, okay, basically they are actually attached, okay, to the carbon, okay, which is the alpha carbon, okay, over here. So the components include, okay, a hydrogen atom, okay, so somewhere here, okay, carboxyl group, okay, somewhere here, okay, amino group, okay, which is here, okay, and also, okay, which is basically straight here, okay, and also variable R group. So this is amino, but this is overall your R group. So like I said earlier on, and in the previous slides, okay, differences in the R group, okay, produce 20 different amino acids. So this is the structure of the amino acids. Now, the 20 different amino acids are further divided into whether it is a non-polar aliphatic or non-polar R groups, or polar but uncharged and charged R groups. So charged R groups, there are only 5. Now, what happens, okay, is that in the non-polar, so as the name says, one group, okay, of the amino acids has hydrophobic R group. Hydrophobic meaning that it is not so water-loving. And another group, okay, the polar, okay, which means that basically they are hydrophilic in nature, so the other way to the non-polar. And the last group, okay, basically they are the one which is electrically charged, okay, and they are either basic or acidic. So the 5 of them, they are further divided into acidic or basic. Now, these are the structures of the 20 amino acids, and don't worry, you don't need to remember each and every one of them, or do you need to remember each of the abbreviation or the short form or the 1-letter abbreviation? No. But again, what I would like you to focus on is basically on the polar, non-polar, okay, and also the special group. So the special group, basically they are from either polar or non-polar. Okay, and what makes them special? If you have ever heard of it, okay, you could let me know, okay, in the comment section. If not, well, you could try to basically we could discuss it, okay, when we meet online, or if you're not interested in it, it's fine, okay. But okay, these three are considered special, and usually the presence of these amino acids in the sequence, especially for bioinformatics purposes, they are basically welcome, simply said, or sought after. Now, take a look, okay, at the structure of each and every one of them. Okay? So cysteine, histidine, glycine, and also proline. So they are basically, even though you can see over here, histidine, okay, is not in one of them, but histidine could be useful. A lot of times it depends on the application. So usually for bioinformatics, histidine is not really sought after, okay, but in lab work, histidine is quite good, okay, because usually we have a his tag for a kind of fusion protein work. So when you look, okay, at the structure, okay, and if you were to look further over here, well, you can refer to your notes. If I'm going too fast, like I said, I'm not going to dwell too much. So those special amino acids, okay, they are categorized differently. Why? Because they basically have a special kind of amino acid attached to them. Amine group, okay, amine group attached to them. So if you were to count each and every one of them over here, you will see that, okay, there are actually 21 amino acids. And even though it even says over here, so okay, the 21, okay, or the 21st, okay, or the latest amino acid is selenocysteine, SEC. So it is being denoted as U. Now, why do we have a special edition? Well, it's actually interesting, but a long story. Okay, so if you're interested, okay, let me know. If not, then we will just move about. So I'm not sure whether it is covered by other lecturer teaching protein before. And apart from the 21st protein, we now have the 22nd protein. Okay, we know that we have selenocysteine. Another one we have is pyrrolysine. So you will see that, again, they basically have a bit of a component or similar to cysteine and lysine, but with an addition in its structure. Again, if you're interested, let me know. Now, further on the amino acids, okay, or the protein, basically they are joined together, okay, by a peptide bond. That is the simplest form of bond joining two amino acids together. So whenever you see, okay, two amino acids joined, usually it is by the peptide bond, okay, nearby bond towards them, and that is the simplest, or basically the kind of like basic form bond formed during the primary structure, which we'll see in a while. So this is repeated over and over to create your polypeptide chain, and it could be very long or very short. It depends on how much, okay, is being attached to each and every one of the amino acid. So at one end of the amino acid, we have a free amino group, okay, which is the N-terminus, and the other is the amino acid with a free carboxyl group, or the C-terminus. So the repeated sequence, okay, would be the one that is making up the backbone of the polypeptide. So attached to the backbone, okay, would be the various R groups. Okay, they are not the one attached to one another. They basically have they do not attach. Okay, basically no bond, okay, will be formed among them unless you are going to have an interaction. For example, protein-protein interaction or protein-ligand interaction. So polypeptide range, of course, okay, basically ranges in size from a few monomers to 1,000, depending on how big the protein is going to be. So this is how the polypeptide backbone, okay, would look like. There is a video, there is a link in your notes as well. I think probably I will put it up in e-learning for you guys to view it. So what happens again is that when the polypeptide backbone, okay, is formed, okay, peptide bond, okay, is actually the ones, okay, that link them together. So the R group is basically free to roam about. That is how they basically are twisted here and there to make sure that they basically do not form a bond with one another. So the peptide bond or backbone, okay, is formed, okay, between, okay, the C-terminus, okay, or sorry, the basically the N-terminus, okay, or the C carboxyl, okay, between two different amino acids. Now, again, just to refresh your mind, okay, we have two different kinds of amino acids, okay, polar and non-polar, and of course, okay, some of them basically are divided, okay, into charge, okay, or either basic or acidic. So the structure of the protein, okay, would actually depends and varies depending on how it is folded, okay. But okay, protein structure, there are 4 structures starting from primary. So the primary is made of 2 or more amino acids joined together, and they are linked by polypeptide bond. That much is known because I've basically covered it in a previous slides. So that is your primary structure of a protein, and it could form polymers, whether a short or a long polypeptide chain. So there are in total 5 bonds that you need to know in protein structure. So your first bond is the peptide bond. So as you can see, okay, it could form a long polypeptide chain. So now basically what you have here, okay, is that again all these amino acids, okay, they are linked, okay, and that makes the sequence of the protein itself similar to a gene, okay, in terms of the nucleotide. So as you can see, okay, even a slight change in the primary structure could affect a protein's conformation and ability to function, because it may not fold correctly or it may not fold at all, and if it doesn't fold, usually protein could serve its function. Now, the second structure is of course secondary. So you have 2 different kinds of secondary structure, which is alpha helix and beta pleated sheet. So as you can see, again, between them, again, one is, as you know, alpha helix. If anyone doesn't know helix, then you need to go back, okay, to your basic, because our DNA is basically in helix structure. So beta pleated sheet, okay, is kind of like when you fold a piece of paper, when you fold them, okay. So those are the structure of beta pleated sheet. So as you can see, because you have your primary structure or your amino acids, okay, basically making up the secondary structure, so they are further more or less twisted, okay. So for example, if they form alpha helix, okay, so they will form hydrogen bond, okay, from the hydrogen group, okay, to an extra oxyl group, okay, basically from another amino acids, okay, whereby, okay, strengthening the bond of the secondary structure. Similarly with beta pleated sheet, okay, the same thing, okay, you can see the bond being formed here. So before we move on further, I normally ask this, okay, and it's usually fun to ask this in class, okay, physically, because we can get lots of instant or spontaneous reaction. But since, well, this is online, so I would just like to ask one question. You could answer it in the comment, or you could just remind me again when we meet online. Now, between alpha helix and beta pleated sheet, in your opinion, which has the strongest bond? Try and look it up. You could try and look it up in Google, okay, but well, just let me know later on. Now, the third structure is of course the tertiary, which is in 3D or 3-dimensional. So this is where your protein starts to fold, and at this stage, tertiary, okay, you will have, okay, 3 added bonds, and as you can see, in total you have 5 bonds. So the structure is of course determined by the interaction of the amino acids located far apart from the primary sequence and the bonds formed between them. So apart from having peptide and hydrogen bonds, okay, which make up the primary and secondary structure, we also have weak interaction, okay, such as hydrogen bonds and van der Waals interaction. So remember van der Waals interaction in your chemistry? Again, I'm not going to dwell too much. We have ionic bond, okay, involving negatively charged and positively charged amino acid side chain group, okay. So those 5 amino acids, okay, which are basic and acidic, when they interact with one another, they form ionic bonds. And the last one, okay, the strongest bond among the 5 bonds in protein structure is the disulfide bond. So covalent linkages that may form from the thiol group of 2 cysteine residue, which are oxidized to a disulfide. So this is very unique, okay, only among the basically 2 cysteine residues, okay, which has the S. Alright, so now, even though some of these interactions, okay, for example, and especially van der Waals, okay, are said to be relatively weak, okay, so the strongest bridge, okay, disulfide bridge, okay, if it is formed in the structure of the protein, usually, okay, it would make, okay, the protein stronger, okay. So just one disulfide bridge, okay, in one protein structure is actually enough, okay, to make sure that the protein is actually compact and probably prevent from denaturation or from unfolding. Right, so the last structure that we have is actually quaternary. So of course, okay, primary, secondary, tertiary, and quaternary. So the most complex, okay, protein structure. Now, quaternary is actually not the protein folded from tertiary. They fold some more and whatnot, I guess, because I have that impression from some student when I was asking them what they think quaternary structure is, okay, but it is not. So we know that quaternary structure is actually formed, okay, from 2 or more polypeptide chain, okay, from 2 domains or more than 2 domains and so forth, okay. For example, okay, I'm giving you the structure. This is actually similar structure. This is just, okay, in ball structure, okay, and this is the carbon structure of hemoglobin, okay. We all have hemoglobin. Hemoglobin is made of 4 domains. So we have 2 alpha chain and 2 beta chain. So this is the quaternary structure, and a lot of other proteins are in the quaternary structure as well. So protein could start to function at basically, okay, tertiary, okay, if everything is okay with the conformation and whatnot, and if it basically is going to be formed, okay, or kind of like complex, okay, with other domains, okay, more than 1 or 2 domains, so they will form the quaternary structure, and probably, okay, would have a better role, okay, or much more important role. Now, usually the quaternary structure is the aggregation or the results of the aggregation of 2 or more polypeptide subunits, okay, when they are linked together. So when we see the hemoglobin just now in the previous slides, we will see that basically it is in globular structure, whereby, okay, a collagen, you guys know collagen is actually a protein, right? So collagen is in the form of elastic, okay? So it's actually kind of like fun structure. So if you guys could imagine, okay, for the collagen, okay, take a look at your hair. Okay, so your hair, okay, has a protein called keratin. So basically, okay, that, okay, how collagen, okay, would be represented, okay, the structure. So collagen is, as we know, is a fibrous protein of 3 polypeptides, okay. They are super coiled like a rope, okay. So if you were to kind of like tie your hair in the braided kind, okay, so kind of like that, and this provides the structural strength for their role in connective tissue, and hence why collagen is actually much sought after to keep your skin firm, of course, for women. And hemoglobin, okay, which we know that is needed, okay, to make our blood, okay. So it's a globular protein with the copies of the 2 kinds of polypeptide. We know that 2 beta chain and 2 alpha chain. Now there you have it, okay, all 4 different structure, okay, from primary up until quaternary. And bear in mind, okay, in total, when you get up until here, you get up until 5 bonds being involved. Over here, there's only peptide. Over here, peptide and hydrogen. Over here, you have peptide, hydrogen, van der Waals, ionic, and also disulfide bridges. So similarly with this. Now, there is actually, the link to the video is actually here. I'll try and see whether it's workable or not and put it in e-learning so that you can view it. Alright, so let's move on to protein folding. So if you remember what I said earlier, that protein basically needs to fold correctly for it to serve its purpose, or before it could function properly. So basically the peptide bond, okay, what it does during the protein folding, okay, they are rotated, as you can see in the beta structure, in the beta and beta sheet, and also alpha helix structure, okay, at the secondary. So in the 3D structure or the tertiary, it actually fold or rotate some more. So usually, okay, what happens is that, okay, when protein fold and orient, okay, it's actually the R groups, okay, that are basically rotated, okay, to either form, for example, okay, form disulfide bridges or form some kind of weak ionic bond or the van der Waals interaction. Now, in the bonds, okay, in protein, okay, there are non-covalent bonds, okay, as we can see, okay, ionic, van der Waals, and also hydrogen. So what we meant as non-covalent, okay, is that usually it is not really that strong of a bond, okay, but it's basically the ones, okay, that holds the protein structure together. So again, taking an example, okay, of proteins. So proteins are further divided into globular and elastic protein. So in a globular protein, we know that one of the example is actually hemoglobin, so it's a simple structure. So what happens is that, okay, from the unfolded polypeptide, okay, you will see that how it actually folds, okay, to form the globular structure. So that means that imagine that you have a piece of paper. So paper is basically thin. You crumple it up, okay, making it into a ball. So that is basically the representation of how globular protein is being folded or conformed. Okay, so the side chain, okay, will help determine the conformation in the aqueous solution. So now, what do the hydrogen bonds do in your protein? So if you know your basic chemistry, we know, or basically basic molecular biology, basically hydrogen bonds, okay, it is the ones, okay, that form a general or basic connection, okay, between one amino acid to another. Or in this case, okay, in protein case that is true. Okay, in DNA case, it's actually from one nucleotide to another. So hydrogen bonds, okay, usually are formed between atoms involved in the peptide bond, okay, primary structure, peptide bond atoms, and also other groups, okay, that would be in the tertiary structure. So going back to protein folding, so when protein folds, okay, you will have a sort of shape, okay, being formed. So this is of course, okay, determined by the sequence of the amino acids, because how they fold, basically it depends on the protein structure. So the final shape is called conformation, and of course has the lowest free energy possible. So that means that they don't really require that much energy, okay, to conform, so that it could later on serve its purpose. So another process called denaturation, okay, is the other way around, or the vice versa of the protein folding, which is unfolding of the protein. So we will go to that again in a while. So denaturation of protein could occur, okay, and it could come down, okay, to a few factors, okay, but okay, the most factor that affect, okay, protein denaturation are heat, pH, or chemical compound. We will see, okay, some of them, and if I would have the time, then during online I will actually show you how this occur. So in the chemical compound, okay, so usually it could remove and have the protein renature or refold, which means that, okay, your protein from its tertiary structure is now back either to the secondary or primary structure, but it could refold, because sometimes, okay, certain chemicals could just be eliminated, okay, so that the protein could refold back and serve its function. So this is how, okay, a normal protein, when it is denatured, so basically it's form, okay, a denatured protein or a loose protein, okay, so you could get back, okay, to your primary structure. So if a chemical is involved for protein denaturation, you could actually renature it back, okay, to have the normal protein. Again, okay, it won't probably be 100%, okay, normal protein in terms of its structure, but it could still serve its purpose regardless. So the degree of renaturation and the effect of renaturation will basically play a role in how correctly renaturation will occur. So basically this is what I've been talking about as well. I've actually just summarized it, okay, so never mind, you can just refer to your notes. Now the one that I actually would like to go through is actually on protein refolding. Now everyone knows an egg, right? So, and everyone knows that the egg white is the one, okay, with the protein. But do you know that actually the egg yolk do have, okay, some proteins? It's just not as much, okay, as the white. So we know that the egg white has protein. So what happens when you basically fry an egg? Okay? From the clear egg white, it becomes white, right? So that is denaturation occurring. Can you refold it back? Can you turn back the white of the egg yolk back to the clear structure? So you could, by right you could. Okay? So I mean, logically speaking, you could, but in the lab actually they have tried to reverse the reaction and it could. Okay, so that's one special thing about research. So in terms of refolding, okay, so only certain chemicals, okay, could help with the refolding. That is what I would like to stress about. So when you have a purified protein isolated from the cells, okay, and it is exposed to high concentration of urea, for example, okay, and this is chemical compound, okay, being involved, so the protein would be denatured. And basically we would add urea in a lot of proteomics study, okay, just to denature protein before, especially in 2D, okay, before it is run on the 2D gel. So once the protein has been denatured, usually the urea is removed. So once the urea is removed, usually protein, okay, could fold back. Okay? And again, this is what I meant, okay, so whether it will refold back, okay, to 100% its original, it's usually not so, but it will refold back and usually it could perform its original function. Alright, so I think that would be the end of the first video, so let's move on to the second video.